So let's get started with Alice Berners-Lee from UC Berkeley, who will be talking about replay slows down with experience. All right, go for it, Alice.
All right. Hi, everyone. I'm Alice, and I'll be talking about, how replay slows down with experience. So first, I'm just going to give a bit of background and motivation for these experiments. Then I'll show you that replay appears after a single experience. Then I'll show you how it slows down with subsequent experience by adding detail, and then a little bit of some summary, and if time, some implications. So for background, place cells in the hippocampus represent space. So on the right, showing place fields of 4 different cells in an open arena. And on the left, you can see that if you record from enough cells, place cells cover the entire track. So here, looking at a linear track, our lab records in rats, and all the experiments that I'm going to tell you about today are rats running back and forth on a linear track from one reward to another reward back and forth. So because we have the encoding model of place fields, we can use really simple Bayesian decoding, memoryless, no prior, to decode the animal's position at any one time during behavior, because we have the place fields of all the neurons, and then we have when they're spiking. So you basically can decode where the hippocampus thinks the rat is at any one moment in time. So this means that it's possible to accurately decode where the animal is while he's running across the track. So here I'm just showing a schematic of those about 100 neurons that were recorded simultaneously. And you can see that as the animal runs across the track, the rat's actual position is in blue. You can see that each cell fires in a different location across the track. And if you look at the decoder estimate of position, so this is high probability in white, low probability in black, you can see that you can accurately track where the animal is as he's running across the track. Then when the animal gets to the end of the track, stops running, starts to drink the reward, what you see is that these cells seem to fire in bursts of activity. And so if you use a shorter timescale decoder to decode what the spatial information is within these bursts of activity, then you can see that they are not actually all firing all at once. They're firing in these sequences where the cells fire in the order in which they fire during behavior. And so you can decode that as this trajectory that goes across the track. And this is associated with a sharp wave ripple in the LFP around 150 to 250 hertz. So, we think that these replays are really cool, and they could be important for a lot of things, including maybe memory. If you disrupt sharp wave ripples, you see deficits in learning. And, it would be very advantageous to be able to replay experiences in order to consolidate those memories for later decisions. So one question we have is, how flexible is replay? For instance, the hippocampus is thought to be important for episodic memory. Could replay also be supporting those types of functions? Episodic memories happen one time. They're not like when a rat is running up and down across the track over and over again, which is usually the way that we look at replay. So we wanted to ask whether replay would appear after a single experience. And so to look at that, I looked at data recorded by Brad Pfeiffer, a former postdoc in the lab. And what he did was he allowed a rat to sleep in a familiar rest box before a run, then placed the rat on a novel track and allowed the rat to only run either 1 or 2 passes across the track. And then took the rat off and put it in the same familiar sleep box. And so the way that we identify replays is with 2 criteria. So this is a nice replay. You can see it has a trajectory across the track. This is the decoded posterior, so the position on the y-axis and the time on the x-axis. And here are 2 other events. So we're looking at just any bursting activity where there's lots of hippocampal neurons firing at once. And these don't look like replays here. And so the 2 criteria that we use to pull out good replays are the weighted correlation, which will be high when the posterior is tight and it's traveling in a direction across the track. And then the max jump distance in between time bins as a proportion of the track. So in between each time bin that is decoded, how far is the replay moving? And so this event does not pass criterion because it has a low weighted correlation, even though it has low max jump distance. whereas this event seems to just, represent one side of the track and then have a huge jump to the other side of the track. So that wouldn't qualify as a replay here. It's a large max jump distance. So this is how we pull out good replays. And if we look at, say for one session, the 3 best events in this pre-task rest, these are what these 3 best events look like. So they don't, look like good replay. But after just one pass across the track in this session, you see nice replay events that pass our criteria to be replay. And if I look across all of the rats that Brad recorded, what you see here is what I'm doing is comparing all of the, so for each session, I take the number of replays that actually happen, compare it to a shuffle distribution where the time bins of each replay are shuffled. And so what you're seeing here is a p-value for each of these across both of these dimensions of our criteria. So max jump distance and weighted correlation. So nice replays are in this green box. And what you can see is that in this pre-rest period, there's no more replay than you would expect by chance. But after just one lap on a novel track, you see that there's robust replay that appears after this single experience. So this was really cool to see. And then the next question we wanted to ask was, after that first experience on this novel track, does replay change as the animal gets more experience in that environment? And so this is data that it was collected by a former graduate student in lab, Delia Silva. And what I'm showing here is the behavior, an example session. The rat is running up and down this track and pausing at the ends. And I just pulled out 7 different example replays that passed our criteria to be replays. On the top, I'm showing the decoded posterior, so the position across time. And on the bottom, the spiking of the hippocampal cells that went into that posterior, ordered by where the place field is on the track. And so what I hope you can see here is that as the animal takes laps across the track, the replays start off rapid, really short in time, and they just dart across the track. And then with experience, these replays get longer and longer. And we looked at across all the animals in dozens of sessions, and saw that replays do increase in their duration and decrease in their slope with experience. So these are all on novel tracks. And I won't show you this today, but I've narrowed this down to local replays that are significant in an environmentally novel experience. So one thing I wanted to look at is, when I was thinking about the mechanism that could support this, is whether the experience is really what's necessary to do the slowing down, or whether being in a novel environment just triggers some process that happens on its own in the hippocampus. And so to look at that, I looked at the replays during this rest period that Brad recorded to see whether after a single pass on a track, the replays were slowing down on their own. And what I found here, I'm looking at just the duration across the period during the rest period, and I found that they did not change. Their slope did not change either. Then when you put the rat back on the same novel track that it's only experienced one pass on and let it explore this novel track, then the replays start to get longer and slow down. So it really does seem that it's the experience of the laps, not just time, that is making these replays slow down. Another thing about replays is that they don't move smoothly across the track. They actually jump and hover in these steps across the track. So I also wanted to look at how replays slow down at this time scale. And so they slow down locked to this slow gamma rhythm that's about 25 to 50 hertz. And what I found is that replays slow down by adding smaller steps. So these jumps and hovers across the track that are thought to maybe be a signature of like underlying attractor dynamics don't seem to change in their frequency or any of the temporal aspects of them, but the replays just add more of these steps on. So in this schematic, you can see in the beginning, there's 3 big steps across the track, which is how the replay gets across the track. But then in later replays, you could have 5 smaller steps across the track. So across all of our replays, the number of steps goes up and the step size decreases across laps in a novel experience. And stuff that I won't tell you here, we actually think that the addition of these hover points where the replays hover themselves are experience dependent. There are fewer hover points than you would expect on the first lap. So it may be that you need to have experience in order to insert these hover locations into these replays. So in summary, what I told you today is that replay appears after a single experience and it lasts irrespective of where the rat is. I did some other experiments to look at that. That significant replay of a local environmentally novel space slows down with experience, and that replays slow down by adding detail to the representation in these hover points. And so I think that this could be, this could make sense as a good mechanism to have when you first are in a novel environment. You want to have quick representations that get you across the track to review, the safe spaces, to update your values of the reward that you just got in this environment quickly. And then later with experience, you may want to slow that representation down in order to add detail. Any downstream area that is listening to these representations would be able to have more detailed representation of experience to maybe build on some cortical or other schematic representation on top of that, or to be able to update states with more granularity. Yeah, so in summary, I want to acknowledge all the people whose data went into this work. Ting Feng did a lot of the early data organization. Delia Silva did the novelty data. I didn't get to show this PFC work that went in, but Jiajing Wu did that. Ellen Ambrose did this reward manipulation to test whether reward would change replays, which we found did not, which I didn't get to tell you about. And then Brad Pfeiffer did the single lap experiment. And that's all. Thank you all for listening.
Great, ended right on time, perfect. Thank you for that excellent talk, Alice. We already have quite a few questions, so let's get into that. Remember, we have 3 minutes. First is from Eleanor Duvel. Did you look at interneuron activity and could they contribute to slowing down?
Yeah, so I sort of skipped that just due to time. But yeah, we do think that they could contribute to the slowing down. It might be that they are less active in the beginning of experience. And I have this sort of idea that, the connections between the pyramidal neurons are not as strong in the beginning because you haven't had, theta sequences across the track or you haven't had replays. But because there's a low interneuron drive, you are still able to have these jumps from these locations and these rapid replays. But then as the connections get stronger and inhibition goes up, you need stronger connections in order to have, in order to get across the track. But you have that because you've had theta sequences, you've had behavioral timescale plasticity and replays in order to get those connections to be stronger. So yeah, we think that inhibition may be a good potential mechanism.
That's a great answer, thank you. So the next one is from Aditi Bishnoi. Sorry if I butcher any names. What is the length of the track? How many cells is it allowed to record in a single lap? And how do the number of cells affect your replay analysis?
Cool, great question. So the data I showed today, they're all from about 1.2 to 1.5 meter tracks. There's not that much variety between them. It would be really cool to look at this in a larger environment and see replays slowing down like really long replays slowing down. And yeah, we believe that getting the most cells is really what we're always trying to do. And so sessions where there aren't as many cells is usually around 100 or so neurons recorded simultaneously to 200 to, there's a lot of variability, but sessions where you have fewer than maybe like 50, it's really hard to get replay. And so in the analysis that I was showing, all of that I was showing were only significant replays, and you really don't see a lot of significant replays when you get down to something like 30 or 40 neurons, unless you get really lucky about the way they are. But yeah, so we have to have a lot of neurons simultaneously.
All right, great. And let's see if we can answer this last one really quick. Eleanor Duval says, nice talk. Have the rats experience another track before the novel track? And do the one-shot replays also represent that other track?
Yeah, so all the rats have experienced another track before. And yeah, there definitely are like remote replays of other tracks. And that's also something that I looked at when I was seeing whether this effect was about the actual like gaining of experience. I looked at on a novel track where I already have, where the animal was just on a different novel track. And I can see whether the replays of the local environment slow down, which they do, but the replays of that first novel track don't slow down while the animal is on this second novel track. So they exist. There are these remote replays, but they stay the same as they did while like after the animal got off that track, which is consistent with the idea that you actually need to be gaining experience in order for these replays to slow down.
All right, great. Great. Thank you so much for that.